Friends, welcome to Sankalp Learning Classes. Today, we are discussing the very important questions for your board exam from this chapter Molecular Basis of Inheritance. Already, we discussed the MCQ questions and some important 2-mark questions. In this session, we are discussing the 2-mark questions as well as 5 marks questions. Okay, so let us start with the first question. Here, you know that I repeat once again, from this chapter total 13 marks questions will be asked and one MCQ and two 2-mark question and the 5-mark question. 5-mark questions, so that is two total 10 marks from the 5-mark session and 2-mark question and 1 MCQ total 13 marks. Okay. So one of the very important chapter for your board exam. So, in this session, we are discussing the expected 5-mark question and very important and previously asked questions. So, like that, we are discussing the questions. Let us start with the 2-mark questions. In the last class, we have discussed the few 2-mark questions. So, let us continue that. What is the cause of discontinuous synthesis of the DNA of one strand on one of the parental strand? What happens to these short stretches of the synthesized DNA? Here, during the DNA replication, just I will take uh, this as a DNA strand, which is having the 5 prime to 3 prime direction, and it is also a 3 prime to 5 prime direction. Okay, here the DNA replication, that is begins so in the direction of 5 prime to 3 prime direction. Always the the nucleotide, so that adds in the direction of 5 prime to 3 prime, means so this strand that adds the the DNA polymerase that add the nucleotides in the 5 prime to 3 prime direction. So this is the continuous strand. But in for this strand, so this is already having the 5 prime to 3 prime to 5 prime direction. So the DNA polymerase cannot add the nucleotides here. So the small RNA primer is added here. So that provides the so by using that RNA strand, the DNA polymerase adds the nucleotide. Again, there is a addition of the RNA primer. After that, there is a adding of the nucleotide. So it looks like it is called as a discontinuous strand. So here, what is the question? What is the cause of discontinuous synthesis of the DNA of one strand on a of a parental strand, one of the parental strand? Here, during the process of replication of DNA, each of the parental DNA strands used as a template. Both strands are used as a template. The strand which is elongating from 3 prime to 5 prime direction will grow continuously, but the strand which is growing from template having the polarity 5 prime to 3 prime direction will grow discontinuously, because RNA primer will be formed at the 5 prime end only. So the discontinuous strand will require many RNA primers. So the discontinuous strand that require many RNA primers, and the strand will be synthesized in small segments of Okazaki fragments, and after that these fragments are removed and the enzyme ligase that binds all the nucleotides and form the another strand. So this is the answer for this question. Next question: how deoxyribonucleoside triphosphate serve dual purpose in a replication? Deoxyribonucleoside triphosphate. So here this deoxyribonucleoside triphosphate that serve as dual purpose in a DNA replication by acting as both a substrate and a energy source. Both a substrate which act as a substrate which nucleotides are added, and the energy source. Next question. Draw a clover leaf model structure of tRNA. So this question, so that is one of the important question. So this is the clover leaf model of the tRNA, which contain the arms like DHU loop, anticodon loop, and T-Cy loop, and here another one, the H loop ಅಲ್ವಾ. ಸೊ ಈ ಟಿ ಆರ್ ಎನ್ ಎ ಡೈಗ್ರಾಮ್ ಅನ್ನ ನಿಮ್ಮ ಬುಕ್ಕಲ್ಲಿ ಕೊಟ್ಟಿಲ್ಲ, ಬಟ್ ದೇ ಕ್ಯಾನ್ ಆಸ್ಕ್ ದಿಸ್ ಕ್ವಶನ್. ಡ್ರಾ ದ ಸೊ ದಿಸ್ ಇಸ್ ಕಾಲ್ಡ್ ಎಸ್ ಎ ಕ್ಲೋವರ್ ಲೀಫ್ ಮಾಡೆಲ್ ಆಫ್ ಟಿ ಆರ್ ಎನ್ ಎ. ಅಲ್ಲಿ ನಿಮಗೆ 3D ಮಾಡೆಲ್ ಅನ್ನ ಕೊಟ್ಟಿದ್ದಾರೆ. ಸೊ ದೇ ಡಿಂಟ್ ಗಿವ್ ದ ಕ್ಲೋವರ್ ಲೀಫ್ ಮಾಡೆಲ್, ಬಟ್ ಇಂಪಾರ್ಟೆಂಟ್. ಸೊ ಯು ಕ್ಯಾನ್ practice this. What is the contribution of Friedrich Miescher in the molecular biology? Here, the scientist who discovered first DNA, but at that time it is not considered as DNA, which is not named as DNA. So DNA as an acidic substance present in the nucleus was first identified by the scientist Friedrich Miescher in the year 1869. He named it as a nuclein. He named that acidic substance which are present in the nucleus, that is named as nuclein, and he discovered that molecule we know and refer to as a DNA, and the some techniques for extraction for it. Okay, so this is the very important concept where the DNA identification begins. So the first scientist who identified the DNA, but at that time period it is not named as a DNA, so he called it as a nuclein. Okay, so next question. The sequence of coding strand of DNA in transcription unit mentioned below. Write the sequence for its complementary strand and its mRNA. So what is the complementary strand for this strand of DNA? That is, I will write 3 prime what it is here. A T G C A. Just a minute. So the complementary strand that is having the 5 prime to 3 prime direction. Here adenine that is bind with the A T here A G C alva C G A T G C C. So cytosine binds with the ATCG alva ATCG. So here CGGC. T, thymine binds with the adenine. Then AT ATCC. So this is the complementary strand. So ಇಲ್ಲಿ ನೋಡಿ ನಿಮ್ಗೆ ಇದು ಗೊತ್ತಿರಬೇಕು. Adenine always binds with the thymine ATCG. Cytosine binds with the guanine with the 3 hydrogen bond. ಗೊತ್ತಾಯ್ತಾ? ಸೊ ಇದು ಗೊತ್ತಿದ್ರೆ ನೀವು ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಸ್ಟ್ಯಾಂಡ್ ಅನ್ನ ಬರಿಬಹುದು. ನೋಡಿ DNA one strand ಗೊತ್ತಿದ್ರೆ, so this is the one strand, so you have to write the another strand. So another strand they have the another nitrogen base alva. So that is the correct answer. And on the mRNA that is always having the 5 prime to 3 prime polarity. So ಇಲ್ಲಿ ಏನಾಗಿರುತ್ತೆ, ಅಡಿನೈನ್ ATCG ಅಂತ ಇದೆ ಅಲ್ವಾ. So this adenine that binds with the thymine, so that is which is not binds with the thymine, that binds with the uracil. Okay, so ಇಲ್ಲಿ ನೋಡಿ adenine ಇದೆ, so that is binding with the uracil U So thymine is given. So adenine, then G, so that is C, then G, U, A, and guanine binds with the cytosine. Cytosine binds with the guanine. Then A, again U, then G, cytosine, then C, G. So ಈ ತರ ಬರಿಬೇಕು. Only what you have to do, thymine that should be replaced by the uracil. U ಅಂತ ಬರಿಬೇಕು. Let us see the answer. Just a minute. So thymine, sorry, adenine binds with the ATGC ಅಲ್ವಾ? Adenine binds with the thymine and guanine binds with the cytosine ಅಲ್ವಾ? So thymine, just a minute. Here the mRNA that is always 5 prime to 3 prime direction, and here the mRNA is having the ಮತ್ತೆ ಸೇಮ್ ಇರುತ್ತೆ ಅಲ್ವಾ, 3 ಪ್ರೈಮ್ ಟು, ಸಾರಿ ಜಸ್ಟ್ ಅ ಮಿನಿಟ್, ಇಲ್ಲೇನಾಯ್ತು ಅಂದರೆ ಐ ಹ್ಯಾವ್ ರಿಟರ್ನ್ ದ ಎ ಟಿ ಜಿ ಅಂತ ಇತ್ತು ಅಲ್ವಾ. ಸೊ ದಿಸ್ ಈಸ್ ಆಲ್ರೆಡಿ ಎಮ್ ಆರ್ ಎನ್ ಎ, ಯಾಕೆಂದರೆ ಒಂದು ಸ್ಟ್ಯಾಂಡ್ ಏನಾಗುತ್ತೆ, ಸೊ ದಿಸ್ ಸ್ಟ್ಯಾಂಡ್ ಆ್ಯಕ್ಟ್ ಆಸ್ ಅ ಟೆಂಪ್ಲೆಟ್ ಸ್ಟ್ಯಾಂಡ್ ಥರ ಆ್ಯಕ್ಟ್ ಆಗುತ್ತೆ. ಸೊ ಇಲ್ಲಿ ನೋಡಿ ಎಮ್ ಆರ್ ಎನ್ ಎ ಏನಿರುತ್ತೆ ನೋಡಿ, ಇಲ್ಲಿ ಥೈಮಿನ್ ಪ್ಲೇಸಲ್ಲಿ ಯುರಾಸಿಲ್ ಇರುತ್ತೆ ಅಷ್ಟೇ. ಸೊ ಓನ್ಲಿ ಥೈಮಿನ್ ಪ್ಲೇಸಲ್ಲಿ ಯುರಾಸಿಲ್ ಇರುತ್ತೆ. ಸೊ ರಿಮೈನಿಂಗ್ ಆಲ್ ಆರ್ ಸೇಮ್ ಅಲ್ವಾ. ಯಾಕೆ ಸೇಮ್ ಇರುತ್ತೆ? ಸೊ ಬಿಕಾಸ್ ಒನ್ ಸ್ಟ್ಯಾಂಡ್ ಆಕ್ಟ್ ಆಸ್ ಅ ಟೆಂಪ್ಲೇಟ್ ಸ್ಟ್ಯಾಂಡ್, ಅನದರ್ ಸ್ಟ್ಯಾಂಡ್ ಆಕ್ಟ್ ಆಸ್ ಅ ಕೋಡಿಂಗ್ ಸ್ಟ್ಯಾಂಡ್ ಅಲ್ವಾ. ಸೊ ಅಲ್ಲಿ ಇನ್ನೊಂದು ಸ್ಟ್ಯಾಂಡ್ ಏನಾಗಿತ್ತು, ಕೋಡಿಂಗ್ ಸ್ಟ್ಯಾಂಡ್ ಆಗಿತ್ತು. ಸೊ ದಿಸ್ ಸ್ಟ್ಯಾಂಡ್ ಥೈಮಿನ್ ಇದ್ದಲ್ಲಿ ಏನಾಯಿತು, so already this strand is synthesized. So A adenine, so that binds with the thymine, that is the uracil alva. Uracil ಗೆ replace ಆಗಿದೆ. Then thymine, so that is replaced by the uracil. Same ಇದೆ ಅಲ್ವಾ. So just I got confused, but this is the correct answer. I hope you got the answer. Sorry for the inconvenience. Next question. Retrovirus do not follow the central dogma. Comment on this statement. So you know that central dogma of molecular biology, that is DNA, so that transcribe into a mRNA, and mRNA that translate into a protein. So this is the central, that is the rule of the central dogma of molecular biology. But here that retrovirus do not follow this rule, because the retrovirus that have RNA as their genetic material and use the reverse transcriptase to convert it into a DNA. Means here RNA is converted into DNA by the reverse transcriptase enzyme. Means here DNA is not synthesizing the RNA, but RNA is synthesizing the DNA. So this process is called as a reverse transcription, because it goes opposite to the direction of the central dogma. So in the opposite direction it goes. So it is called as a so the retrovirus do not follow the central dogma of the biology rule. Next question. Write the criteria for molecule that can act as a genetic material. Here, the molecule should contain these characteristics. It should be able to generate its replica. It should undergo the replication. It should be stable chemically and structurally. It should provide the scope for slow changes. So there should be a mutation so that are required for the evolution. It should be able to express itself in the form of Mendelian characters. So these are the characters that should the molecule that should act as a genetic material that should perform. Okay. 2 mark question, one of the very important question. Write the difference between heterochromatin and euchromatin. ಇದನ್ನ ತುಂಬಾ ಸಲ ಕೇಳಿದ್ದಾರೆ ಈ ಕ್ವಶನ್ ಅನ್ನ. Write the difference between the heterochromatin and the euchromatin. Here the nucleus, a typical nucleus, some region the chromatin are loosely packed and are referred as euchromatin. If the chromatin are loosely packed, they are called as euchromatin. But the chromatin that are densely packed and stains dark are called heterochromatin. But here the euchromatin that is lightly stained, heterochromatin that is darkly stained. And euchromatin that is said to be transcriptionally active chromatin. Euchromatin is the transcriptionally active chromatin, but heterochromatin that is a inactive chromatin. So these points you should write for the difference between the euchromatin and the heterochromatin. So again I repeat once again, make the 2 columns and write the euchromatin and heterochromatin and write the difference, okay, for 2 marks. Draw a neat label diagram of nucleosome. So this question is also a repeated question. This is the structure of the nucleosome where histone proteins are there, and the DNA that is wrapped around the histone octamer. So that is a nucleosome, very important diagram, and practice the labelings also. Then next question. Mention the polarity of the template and coding stand of the transcription unit. Here, this is the transcription unit. Here you should mention so this is the template stand and this is the coding stand. You have to mention, just they ask to mention the polarity, but I had given the diagram here for your understanding purpose. So the template stand have the 3' to 5' polarity, and coding stand have the 5' to 3' polarity. Here the template stand that act as a stand that should produce the new mRNA, always mRNA, because of that, which is having the 5' to 3' direction polarity. Okay. So these are the some 2-mark questions, very important 2-mark questions. Next, we are discussing the important 5-mark questions. Okay. So next, the very important topic, 2 5-mark questions will be asked. That is about the 10 marks. Alva. So 10 marks ಏನು ಬರುತ್ತೆ ಇಲ್ಲಿ ಬರುತ್ತೆ ನಿಮಗೆ. So let us discuss the first question. DNA replication is fast, accurate, energetically expensive, substrate and enzyme dependent, initiated from the specific site and cannot uncoil on its entire length. Explain the process of DNA replication considering all these features. By using these all features, you have to explain the DNA replication. ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್ ಟಾಪಿಕ್. ನಿಮಗೆ ಡೈರೆಕ್ಟ್ ಆಗು ಕೇಳ್ಬೋದು DNA replication ಅನ್ನ ಬರೀರಿ ಅಂತ. Otherwise they can ask the question like this. So at last you have to you have to explain the DNA replication. Okay, one of the very important topic. Nodi. So first step, DNA replication is a crucial biological process that ensures the accurate duplication of the genetic material. It occurs in several steps. So it involves the enzymes and substrates. The process that begins at the specific site of the DNA molecule known as a origin of the replication. It originate at a specific site, that is called as a origin of a replication, and it is called as a semi-conservative manner, whereas the each new DNA molecule consists of one original strand of the newly synthesized strand, one original strand and one newly synthesized strand. Because of this, it is called as a semi-conservative. Next question. The first step is the DNA replication start at the specific locations called the origin of replication. Here, the protein recognize these sites and bind them, so that enzyme that is called as a DNA helicase enzyme, DNA helicase enzyme, that perform the unwinding the DNA strand, so that will forming the um, so yes, here unwinding the DNA double helix by the enzyme helicase. Yes, so the helicase enzyme that unwinds that separate the two strands by removing the hydrogen bond between the two strands. So now it is called as a replication fork. So how it is formed? This is the DNA strand. Just imagine this is the DNA strand. So this hydrogen bonds are removed. So it looks like this. So like this. So this is the unwinded DNA strand, and here the helicase enzyme is, so that is removing the hydrogen bonds. So this is called as a replication fork. The unwinding of the DNA creates replication fork, whereas two strands of the DNA are separated and allowing the access for the replication process. Now the replication process starts. Then RNA primers. These RNA primers, they are synthesized by the RNA primase enzyme. So RNA synthesizes the short RNA primers. So they are used as a complementary to the DNA template strand, and these primers provide the starting point for the DNA synthesis, for the synthesis of the another strand. Here the DNA polymerase enzyme add the nucleotide. It start to add the nucleotides to the growing DNA strand using the original strand as a template. So this process is fast, occurs at the 5 prime to 3 prime direction. The DNA uh, polymerase enzyme that adds the nucleotides in the direction of the 5 prime to 3 prime direction in a one strand, but this creates the some additional complication in the replication. So here one strand that is having the 3 prime to 5 prime polarity, the replication is continuous. The ಇಲ್ಲಿ ನೋಡಿ. So this is the 3 prime to the 5 prime strand. So in this strand, just a minute. Here 3 prime to the 5 prime strand. So here the replication is continuous in the 3 prime to the 5 prime strand, because the replication start at the 5 prime end, so it continues to the 3 prime end. But another strand which is having the polarity 5 prime to 3 prime, so that is that synthesis the new strand that is called as a discontinuous, because here the RNA primers they are required to initiate the replication. So RNA primers ಅನ್ನ ಇಲ್ಲಿ replication ಇನ್ನೊಂದು ಸ್ಟ್ಯಾಂಡ್ ಅನ್ನ ಸಿಂಥಸಿಸ್ ಮಾಡಲಿಕ್ಕೆ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ಅನ್ನ ಆಡ್ ಮಾಡಲಿಕ್ಕೆ ಇಲ್ಲಿ ಯೂಸ್ ಮಾಡ್ಕೊಳ್ತಾರೆ. So which contain the RNA primers, so it is called as a discontinuous strand. Then after the synthesis, the DNA polymerase have the proofreading abilities. If any error occurs during the replication, so it ensures high of fidelity and DNA replication. So which identify the errors and which correct the that errors. And termination. So the replication continues until the entire length of the DNA so that DNA molecule has been copied. So specific termination sequences signal are at the end of the replication, and the DNA ligase which binds the new strand and which form the new strand in the DNA replication. Okay, so this is the process of the DNA replication. So one of the very important question, repeatedly asked question in your exams. Okay, next question. This is also a model question paper question. Given below are the sequences of nucleotides in a particular mRNA and amino acids coded by it. Here 5 prime to 3 prime and AUG, UUU, UUC, GAG, UUA. ಸೊ ಈ ತರ ಏನಾಗಿದೆ, ಸೊ ಇಲ್ಲಿ ಅಮೈನೋ ಆಸಿಡ್ಸ್ ಕೋಡ್ ಅನ್ನ ಕೊಟ್ಟಿದ್ದಾರೆ. ದೆನ್ ಅಮೈನೋ ಆಸಿಡ್ಸ್ ಆಲ್ಸೋ ಗಿವನ್ ಹಿಯರ್, ಮಿಥಿಲಿನ್, ಫಿನ್, FeHe, ಗ್ಲುಟಾಮಿನ್, ವ್ಯಾಲಿನ್ ಈ ತರ ಕೊಟ್ಟಿದ್ದಾರೆ ನೋಡಿ. ಹಿಯರ್ write the properties of the genetic code that can be correlated from the above given data. Write the properties of the genetic code that can be correlated from the given data. Here you have to observe this and find out the characteristics of the genetic code. Genetic code characteristics ಏನಿರುತ್ತೆ ಅದನ್ನ find out ಮಾಡಬೇಕಾಗುತ್ತೆ. So you have to write. So just observe this genetic code. They are triplet in nature. The codon is triplet. Some amino acids are coded by more than one codon, hence the code is degenerate. They are degenerate in nature. The codon is read in mRNA continuous fashion. There is no punctuations. So ಅಲ್ಲಿ continuous ಆಗಿ U ಅಂತ read ಮಾಡ್ತಾರೆ. There is no punctuations ಅಲ್ವಾ? There is no punctuations. The code nearly universal. So that UUU would be code for phenyl alanine. So that is represented at PHE. So UU andre that is a phenylalanine. That is the universal. AUG has the dual function. It codes for the methionine and it is also as a initiator codon. AUG is the initiator codon as well as which codes for the methionine. And UAA, UAG, UGA are the stop terminator codon. They stop the that translation process. So UAA, UAG, UGA, these three, UAA, UAG and UGA are the terminator codons. So these are the characteristics of the these codons. Next question. So read this question. Identify the polarity of X and Y above the given diagram, and how many more amino acids are expected to be added to this elongating the polypeptide chain. Which sequences of bases are generally found at the X and Y polarity? Okay. So ಇಲ್ಲಿ ನೋಡಿ, here this is the mRNA. mRNA always having the polarity 5 prime to the 3 prime polarity alba. So 5 prime to 3 prime polarity irutte. Then how many more amino acids can be added here? So here this UAA, that is the stop codon. UAA enide alva. So that is the stop codon. So we cannot add here. UAA is the stop codon alva. ಇಲ್ಲಿ ನೋಡಿ, UAA, that is the stop codon. So we cannot add another amino acid here. So no amino acid can be added here. ನೋಡಿ ಗೊತ್ತಾಗ್ತಾ ಇದೆಯಾ? So the polarity is 5 prime to 3 prime and no amino acids can be added here. So see the next question. Mention the anticodon for alanine and valine amino acids based on the diagram. ನೋಡಿ, Here alanine, that is having the codon GCA. Alanine, which is having the codon GCA, and valine. This is the valine that is having the codon GUU. So you should write the anticodon. Anticodon ಹೇಗ್ ಬರಿತೀರಾ? Anticodon ATCG ಅಲ್ವಾ? G binds with the C and G, that is T ಅಲ್ವಾ? Then here GUU ಅಂತ ಇದೆ. Here guanine that binds with the cytosine, U that is uracil, A ಅಲ್ವಾ? So these are the anticodon. Anticodon ಯಾವ ತರ ಇರುತ್ತೆ ನೋಡಿ. So these are the codons and these are the anticodons. What is the composition of the catalyst involved in the peptide bond uh, formation in this process? So here these are the amino acids. So these amino acids are joined by the peptide bond ಅಲ್ವಾ? Peptide bond ಅಲ್ಲಿ ಅದು ಏನಾಗುತ್ತೆ? ಜಾಯಿನ್ ಆಗುತ್ತೆ. Here, so the enzyme that binds this amino acid, the enzyme which helps to bind the amino acids, you have to find out for this question. Okay, so this is the model question paper question. Find the answer for this. So what is the composition of the catalyst that is involved in the peptide bond? The enzyme is involved in this, which enzyme, and what is the characteristic of, what is the composition, sorry, what is the composition of the catalyst involved in this peptide bond formation? Okay, that is for your work. Next question. List the silent features of human genome. Yes, so list the silent features of the human genome. Very important question. 5 marks ಗೆ ತುಂಬಾ ಸಲ ರಿಪೀಟೆಡ್ ಆಗಿ ಕೇಳಿರುವಂತಹ ಕ್ವಶನ್ಸ್. The human genome contains 3164.7 million base pairs. The average gene consists of 3000 bases, but sizes vary greatly, with largest known human genome being dystrophin at 2.4 million bases. The total number of genes estimated, that is 30,000, much lower than the previous estimate. Almost 99.9% nucleotide bases are exactly the same in all people. So almost all the people, 99.9 people, so they are the having the same genes. The functions are known for over 50% of the discovered genes, but 50% of the remaining gene functions are unknown. Less than 2% of the genome codes for the protein. Only less than 2% of the protein codes for the protein. Repeated sequences make up very large portion of the human genome. There are repeated sequences are present, and repetitive sequences are stretches of the DNA sequences that are repeated many times, sometimes 100 to 1000 times. They are thought to be thought to have no direct coding functions, but they shed light on the chromosome structure, dynamics, and the evolution. And one more important, chromosome 1 has the most genes, that is 2968, and the Y has the fewest, that is 231. And scientists, next feature, that is the scientists have identified about 1.4 million locations, whereas single base DNA difference, that is a single nucleotide polymorphism pronounced as SNPs, occur in humans. So these all features, so these questions will be asked for 5 marks alva. 5 marks ಗೆ ನೀವೆಲ್ಲ features ಅನ್ನ ಬರಿಬೇಕಾಗತ್ತೆ. So these features carries the ಸೊ ದೀಸ್ ಆಲ್ ಫೀಚರ್ಸ್ ಕ್ಯಾರೀಸ್ ದ 5 ಮಾರ್ಕ್ಸ್. ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್. ಎಲ್ಲ ಪಾಯಿಂಟ್ಸನ್ನು ಕರೆಕ್ಟಾಗಿ ನೋಟ್ ಮಾಡಿ ಬರ್ಕೊಳ್ಬೇಕು. Describe the steps involved in DNA fingerprinting technique. This question is also from the model question paper. So what are the steps involved in the DNA fingerprinting technique? So the DNA sample. So the DNA sample to analyze to be first collected. So extraction. The DNA then extracted from the sample by the lysis method. Digestion by the restriction endonuclease. Using the specific restriction enzyme, the DNA fragment cut at the specific site. This is done to obtain the restricted fragment length polymorphism. Then electrophoresis. You know the process of the electrophoresis, where the DNA fragments, they are separated based on their sizes. And the denaturing of the fragment. The DNA fragment, they are immersed in the sodium hydroxide, and denature means separation of the DNA strand will be done here. Single strand DNA will be done here. Then southern blotting means this is the transfer of the DNA strand into the membrane, the nitrocellulose membrane. Next, the hybridization. This DNA fragment is subjected to hybridization with the suitable DNA probe tagged with the radioactive substances. The suitable DNA fragment is so that will bind with the complementary one. Then comparing the sample of the suspect with the evidence using the X-ray films, the DNA sample showing the radioactivity are compared. So these are the steps involved in the DNA fingerprinting. One of the very important question. ಇದ್ರ ಮೇಲೇನು ಕೂಡ ತುಂಬಾ ಸಲ ರಿಪೀಟೆಡ್ ಆಗಿ ಕ್ವಶನ್ಸ್ ಅನ್ನ ಕೇಳಿದ್ದಾರೆ. Next question. Construct a complete transcription unit with promoter and terminator on the basis of the hypothetical template strand given below, and write the RNA strand transcribed from the above transcription unit along with the polarity. Illi nodi. So here we will consider it as a 3' to the 5' direction, and we have to construct the transcription unit alva. So that should contain the 5' to 3' direction, which again A T A G C C G A T A G C G T G sorry A T C G. So this is the transcription unit which contain the, here there is a promoter and it also contain the terminator alva. ನಾನು just rough ಆಗಿ ಹೇಳ್ತಾ ಇದ್ದೇನೆ. Terminator, and this is the structural gene alva. So this is the transcription unit. ನೋಡಿ transcription unit ಯಾವ ತರ ಇದೆ ಅಂತ. So here just 3' at the 3' end promoter will be there, at the 5' end terminator will be there alva. So 3' end ಅಲ್ಲಿ promoter ಇರುತ್ತೆ, and at the 5' end terminator will be there. Then next, the RNA. So you have to synthesize the RNA based on this sequence. I will draw the, so this is the 3' and which contain the promoter, terminator, which act as a template strand, and this strand act as a coding strand. ಇದು coding strand ತರ act ಮಾಡುತ್ತೆ. 5' to the 3' direction ಇದೆ ಅಲ್ವಾ. So already we know how to obtain the RNA. So RNA always that is having the 5' to the 3' direction alva. So adenine ಇದ್ದಲ್ಲಿ RNA ಅಲ್ಲಿ ಏನಿರುತ್ತೆ? So adenine So that binds with the uracil alva. Adenine that binds with the uracil U. Thymine A. Thymine binds with the adenine G. C. C. G. Uracil and G. C. C. G and adenine uracil. Then A. Just a minute. Adenine binds with here thymine. So that binds with the adenine. Then adenine. I got U 1, 2, 3, 4, 5, 6. I will give the number like this 2, 3, 4, 5, 6. Okay. Adenine, uracil, and thymine, ATCG. Alpha. So that is thymine, adenine. Then G, C. Then C, that is G. Then adenine, uracil, thymine. So that binds with the adenine. Then adenine that binds with the uracil and cytosine that binds with the guanine. So this is the um, mRNA strand, but you should write neatly. I hope ನಿಮಗೆ ಹೇಗೆ ಬರೆಯೋದು ಅಂತ ಅರ್ಥ ಆಗಿದೆ ಅಂತ ಅನ್ಕೊಳ್ತೇನೆ ನೋಡಿ. So you have to draw the another strand, that is the coding strand. First coding strand ಅನ್ನ ಬರ್ಕೊಳ್ಳಿ for the 3 mark question. So for the 3 mark question, this is the 3 prime end and this is the 5 prime end, and 3 prime end which contain the 5 prime and 3 prime. So here, so this is the coding strand and this is the template strand. These are called as structural genes. At the 3 prime end, promoter will be there. At the 5 prime end, terminator will be there. Okay. So this is the, and write the codes on this strand. Okay. Adenine, thymine ಈ ತರ ಇರುತ್ತೆ. ನೆಕ್ಸ್ಟ್ ಆರ್ ಎನ್ ಎನ ಸಿಂಥಸಿಸ್ ಮಾಡಬೇಕಾದ್ರೆ ಯು ಹ್ಯಾವ್ ಟು ಟೇಕ್ ದ ಟೆಂಪ್ಲೆಟ್ ಸ್ಟ್ಯಾಂಡ್ ಆಸ್ ಅ ರೆಫರೆನ್ಸ್. ಟೆಂಪ್ಲೆಟ್ ಸ್ಟ್ಯಾಂಡ್ ಅನ್ನ ರೆಫರೆನ್ಸ್ ತಗೊಂಡು ಬಟ್ ಯು ಹ್ಯಾವ್ ಟು ರಿಪ್ಲೇಸ್ 80 ಥೈಮಿನ್ ಅನ್ನ ಏನ್ ಮಾಡಬೇಕು ಯುರಾಸಿಲ್ ಜೊತೆ ರಿಪ್ಲೇಸ್ ಮಾಡಿದ್ರೆ ನಿಮ್ಗೆ ಏನ್ ಸಿಗತ್ತೆ, ಯು ವಿಲ್ ಗೆಟ್ ದ ಆರ್ ಎನ್ ಎ ಸ್ಟ್ಯಾಂಡ್. ಓಕೆ, ಸೊ ದಟ್ ಈಸ್ ದ ಕರೆಕ್ಟ್ ಆನ್ಸರ್. ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್. With a neat label diagram, explain double helical structure of DNA. The double helical structure of DNA that contains the two strands. Here base pairs are there, adenine, thymine, guanine, cytosine, ಮತ್ತೆ sugar phosphate backbone. So these are the sugar phosphate backbone. So this is the very important diagram. Please practice this diagram. And the characteristics of the double strand DNA molecule. It is made up of the polynucleotide chain, whereas backbone is made up of the sugar phosphate and bases project inside. The two chains have anti-parallel polarity. It means one chain that is having the 5 prime to 3 prime direction. Okay, so this is arrow mark, and other is having the 3 prime to the 5 prime. So they are having the anti-parallel polarity. The bases in two strands are paired through the hydrogen bonds, forming the base pairs. Adenine forms the two hydrogen bonds with thymine and opposite strand vice versa. Similarly, guanine is bonded with the cytosine ATGC with the 3 hydrogen bond. As a result, always purines comes to the opposite pyrimidines. This generates the approximately uniform distance between the two strands of the helix. Then the two chains are called right-handed fashion, and the pitch of the each helix is 3.4 nanometer, and there is roughly 10 base pairs in each turn. Each turn that contain the 10 base pairs. So approximately 0.34 nanometer. The plane of one base pair, so that stacks over the double helix. This is the addition to the hydrogen bonds confers the stability of the helical structure. So these are the characteristics of the double helical model of the DNA. ತುಂಬಾ ಸಲ ರಿಪೀಟ್ ಆಗಿ ಕೇಳಿರುವಂತಹ ಕ್ವಶನ್ಸು. Define genetic code. Explain any 5 silent features of the genetic code. We know the silent features of the genetic code here. The genetic code refers to the instructions contained in a gene that tell a cell how to make a specific protein. So how to make the protein, that is confirmed by the genetic code. We know the silent features of the genetic code alva. So starting ಒಂದು question ಅಲ್ಲಿ ನಾವು ನೋಡಿದ್ವಿ what are the features of the genetic code. They are triplet and they are universal and they are AUG is the initiator codon and the stop codons and the terminator codons. So ಇದೆಲ್ಲವನ್ನು ನಾವು ಕೂಡ ಸೆಕೆಂಡ್ ಕ್ವಶನ್ ಅಲ್ಲಿ ನೋಡಿದ್ವಿ ಅಲ್ವಾ. ಸೊ ಐ ವಿಲ್ ಮೂವ್ ಟು ದ ನೆಕ್ಸ್ಟ್ ಸ್ಲೈಡ್ ನೌ. With respect to the lac operon, mention the functions of the following: regulatory gene, inducer, lac Z, lac Y, and lac A. Here, what is the function of the regulatory gene? The regulatory gene, so that is I, codes for repressor protein. So the repressor gene is produced, repressor protein is produced by the regulatory gene. Then inducer, an inducer molecule that regulates the gene expression by disabling the repressors or binding the activators. LAC-Z. LAC-Z is the structural gene and that codes for the beta galactosidase, which break down the lactose into glucose and galactose. And LAC-Y, the structural gene LAC-Y codes for lac permease, and which is transpermeable protein that helps to cell take up the lactose. And LAC-A, the structural, these ZYA, they are the structural genes. LAC-A codes for beta galactoside transacetylase, so which codes for the transacetylase. Okay, so these are the functions of this regulatory gene, inducer, LAC-Z, LAC-Y, and LAC-A. Next question. Draw a schematic structure of transcription unit and explain it. The schematic structure of the transcription unit is like this, which is having the two stands, that is template stand and the coding stand, and the promoter and the terminator alva. So here you have to write the explanation. ಇದನ್ನ ಕೇಳ್ತಾರೆ, ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್ ಯೂನಿಟ್ ಅನ್ನ ಡ್ರಾ ಮಾಡಿ ಅಂತಾನು ಕೂಡ ಕೇಳ್ತಾರೆ. The strand that has polarity 3' to 5' act as a template and another strand act as a coding strand. The other strand which is having the polarity 5' to 3', so that is sequence same as RNA except the thymine at the place of the uracil. Next nodi. The promoter and terminator flank the structural gene in the transcription unit. Promoter matte terminator, so that is present at the structural gene. Promoter is said to be located at the 5' end upstream, and the structural gene and the terminator, here the reference is made with the respect to the polarity of the coding strand. Coding strand polarity ಅಲ್ಲಿ 5' end ಅಲ್ಲಿ ಇದೆ ಅಂತ ಹೇಳಿದ್ದಾರೆ. It is a DNA sequence that provides the binding site for the RNA polymerase. So promoter andre ಏನು? It is a site where the RNA polymerase binds, and it is the presence of promoter in a transcription unit that also defines the template and the coding stands. By switching its position with the terminator, the definition of the coding and template stands could be reversed. The terminator, so that is located towards the 3' end of the coding strand, and it is usually defines the end of the process of the transcription. So the terminator, so terminator that is present at the 3' end of the coding stand. Coding stand 3' end ಅಲ್ಲಿ ಇರುತ್ತೆ, and that terminates the transcription process, that ends the transcription process. Describe the Hershey-Chase experiment to prove that DNA is the genetic material. Hershey-Chase experiment through DNA is the genetic material, DNA genetic material ಅಂತ ಹೇಗೆ ಪ್ರೂವ್ ಮಾಡಿದ್ರು ಅಂತ ಅಲ್ವಾ. ದೇ ಹ್ಯಾವ್ ಟೇಕನ್ ದ ಬ್ಯಾಕ್ಟೀರಿಯೋಪೇಜ್. ಬ್ಯಾಕ್ಟೀರಿಯೋಪೇಜ್ ಅನ್ನ ಏನ್ ಮಾಡ್ತಾರೆ ಅಂದ್ರೆ, ಬ್ಯಾಕ್ಟೀರಿಯೋಪೇಜ್ ದಟ್ ಇಸ್ ದ ವೈರಸ್ ದಟ್ ಕಾಸಸ್ ದ ಡಿಸೀಸ್ ಫಾರ್ ದ ಬ್ಯಾಕ್ಟೀರಿಯಾ. ಈ ಬ್ಯಾಕ್ಟೀರಿಯೋಫೇಜ್ ಏನ್ ಮಾಡ್ತಾರೆ, ಸೊ the bacteriophage attaches to the bacteria and its genetic material then enters into the bacterial cell. ಆ ಬ್ಯಾಕ್ಟೀರಿಯೋಫೇಜ್ ಏನ್ ಮಾಡುತ್ತೆ, ಅದರ ಜೆನೆಟಿಕ್ ಮಟೀರಿಯಲ್ ನ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಸೆಲ್ಗೆ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡುತ್ತೆ. ಸೊ ದೇ use this method, and what they will do, they grew some virus, the medium that contain the radioactive phosphorus ಮತ್ತೆ the medium ಅಲ್ಲಿ ಏನಿರಬೇಕು, radioactive sulfur ಇರಬೇಕು. ಆ ತರ medium ಅಲ್ಲಿ ಏನ್ ಮಾಡ್ತಾರೆ, they grow the bacteriophage virus. Then viruses grown in the presence of the radioactive phosphorus contain the radioactive DNA. ಯಾವ ವೈರಸ್ ಅನ್ನ ಫಾಸ್ಫರಸ್ ರೇಡಿಯೋಆಕ್ಟಿವ್ ಫಾಸ್ಫರಸ್ ಅಲ್ಲಿ ಗ್ರೋ ಮಾಡಿದ್ರು ಅಲ್ವಾ, ಸೊ ದಟ್ ಕಂಟೇನ್ ದ ರೇಡಿಯೋಆಕ್ಟಿವ್ DNA ಬಟ್ ನಾಟ್ ದ ರೇಡಿಯೋಆಕ್ಟಿವ್ ಪ್ರೋಟೀನ್, ಬಿಕಾಸ್ ದ DNA ಕಂಟೈನ್ ದ ಫಾಸ್ಫರಸ್ ಅಲ್ವಾ. DNA ಇಲ್ಲಿ ಫಾಸ್ಫರಸ್ ಇರೋದ್ರಿಂದ ರೇಡಿಯೋಆಕ್ಟಿವ್ ಫಾಸ್ಫರಸ್ ಅನ್ನ ಅಬ್ಸಾರ್ಬ್ ಮಾಡ್ಕೊಂಡಿದೆ, but protein does not. Similarly, viruses grown in the radioactive sulfur contain the radioactive protein but not the radioactive DNA, because DNA does not contain the sulfur. So sulfur ಇನ್ನೊಂದು ಸ್ವಲ್ಪ ಬ್ಯಾಕ್ಟೀರಿಯಾನ ಏನ್ ಮಾಡ್ತಾರೆ, they grown in the radioactive sulfur. They contain the virus contain the radioactive sulfur in their protein, not in the DNA, because DNA does not contain the sulfur alva. So their protein contain the radioactive material. Next, the bacteriophages, they attach to the E. coli bacteria ke they transfer their genetic material. Okay, they infect that bacteria. Then the virus particles were separated from the bacteria by spinning them in the centrifuge. So ಆಫ್ಟರ್ ಏನ್ ಮಾಡ್ತಾರೆ, ಸೆಂಟ್ರಿಫ್ಯೂಜ್ ಮಾಡಿ ವೈರಸ್ ಪಾರ್ಟಿಕಲ್ಸ್ ಅನ್ನ ಸೆಪರೇಟ್ ಮಾಡ್ತಾರೆ. The bacteria which was infected with the virus that had the radioactive DNA were radioactive, indicating that the DNA was the material that passed from the viruses to the bacteria. The bacteria which contain the radioactive DNA that transfer the DNA to the bacteria, the virus which contain the radioactive phosphorus DNA in their DNA. ಸೊ ಅದೇನ್ ಮಾಡುತ್ತೆ, ಆ ಒಂದು ಬ್ಯಾಕ್ಟೀರಿಯಾಕ್ಕೆ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡಿದೆ. ಬಟ್ the bacteria which contain the sulfur, radioactive sulfur in their protein, which is not transferred the radioactive sulfur. So they concluded that DNA is the genetic material, DNA is transferring but not the protein. So this is the Hershey and Chase experiment. So this is the diagram, very important ನೋಡಿ ಯಾವ. So this is the bacteriophage ಅನ್ನ ಗ್ರೋ ಮಾಡಿದ್ದಾರೆ ಸಲ್ಫರ್ ಅಲ್ಲಿ ಮತ್ತೆ, and then in the phosphorus ಅಲ್ಲಿ, phosphate ಅಲ್ಲಿ. But here how it is transferring the genetic material into the bacterial cell, so it is represented. Next question, describe the process of transcription in eukaryotes. One of the very important question, ಇದನ್ನ ಕೇಳ್ತಾರೆ, transcription in eukaryotes. ನೋಡಿ, transcription, so that is the process of formation of the synthesis of the mRNA from the DNA ಅಲ್ವಾ. ಸೊ ದಟ್ ಈಸ್ ಹ್ಯಾವಿಂಗ್ ದ ಪೊಲಾರಿಟಿ 5' to the 3'. ಇಲ್ಲಿ ನೋಡಿ, ಫಸ್ಟ್ ಸ್ಟೆಪ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, RNA polymerase binds with the promoter sequence to initiate the process of transcription, association with the initiation factor, so that alters the specificity of RNA polymerase to initiate the transcription. So transcription initiation, then next step is the elongation, where the RNA polymerase use the nucleoside triphosphate as the substrate, and the polymerization occurs. So the new strand synthesis will occur here. Then termination. Here the rho factor, so that is required to the terminate the process. At as the RNA polymerase proceeds to perform the elongation, so the short stretch of RNA remains bound to the enzyme. As the enzyme reaches the termination region, so the RNA, so that falls off. So that is represents the termination of the process. So after the termination ಏನಾಗತ್ತೆ, HNRNA, so the produced RNA is called as a HNRNA, and this RNA that should undergo the some process that is called as post transcriptional process, capping, ಟೇಲಿಂಗ್ ಅಂಡ್ ಸ್ಪ್ಲೈಸಿಂಗ್ ಅಲ್ವಾ. ಸ್ಪ್ಲೈಸಿಂಗ್ ಅಲ್ಲಿ ಏನ್ ಮಾಡ್ತಾರೆ, ಇಂಟ್ರಾನ್ಸ್ ಅನ್ನ ರಿಮೂವ್ ಮಾಡ್ತಾರೆ ಅಂಡ್ ಎಕ್ಸಾನ್ಸ್ ಆರ್ ಜಾಯಿಂಟ್. ನೆಕ್ಸ್ಟ್ ಟೇಲಿಂಗ್, ಸೊ ಟೇಲಿಂಗ್ ಅಲ್ಲಿ ಏನ್ ಮಾಡ್ತಾರೆ, ಸೊ poly A tail, poly A synthetase is added at the 3' end, and 7 methyl guanosine triphosphate is added at the 5' end. So that forms the complete mRNA. mRNA synthesis ಆಗುತ್ತೆ. So here there is a diagram which is also very important. ಡಯಾಗ್ರಾಮ್ ಅನ್ನು ಕೂಡ ಇನಿಷಿಯೇಷನ್, ಇಲಾಂಗೇಷನ್, ಟರ್ಮಿನೇಷನ್ ಅಂತ ಇದೆ. ಸೊ ಪ್ರತಿಯೊಂದು ಸ್ಟೆಪ್ ಅಲ್ಲೂ ಡಯಾಗ್ರಾಮ್ ಹಾಕಿ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಬೇಕು ಫಾರ್ 5 marks. ಓಕೆ. So these are the some important questions from this chapter. So thank you. We'll meet in the next session.